Lorena aquí, sí. I think we can bring it a little more further front, no? ¿Qué se siente? ¿Qué se siente? Lo digo ahora esto. Es un placer para mí estar hoy en la ceremonia de la inauguración del Congreso Global Summit Neuro 2022. Se abre la sesión. Tiene la palabra don Carlos García Carballo, alcalde de Salamanca.
Muy buenos días, Su Majestad la Reina de Suecia, Su Majestad la Reina doña Sofía, rector de la Universidad de Salamanca, Ricardo Rivero, delegada del Gobierno en Castilla y León, Virginia Barcones, subsecretaria de Sanidad, Dionisia Manteca, autoridades, distinguidos participantes en esta cumbre mundial. Welcome to Salamanca, a city open worldwide, proud of its ancient past that is constantly renovating itself every day, strongly based upon and linked with knowledge and wisdom. We are deeply proud to be the site of this highly interesting event, gathering in our city the most remarkable scientists in the world, joined together in our compromise so as to advance in the research concerning the causes and treatment of neurologic illnesses. Señoras y señores, Salamanca, la ciudad que les acoge, es ciudad patrimonio de la humanidad y cuenta con una continuidad monumental y una calidad artística únicas. Es la ciudad universitaria por excelencia, un faro histórico del saber que busca su espacio en un mundo en constante cambio al que tenemos mucho que aportar. Somos también la ciudad de la concordia. Por encima de los intereses particulares, las administraciones e instituciones sabemos anteponer el interés general de los salmantinos, que son el motor de nuestras acciones y decisiones. Las personas y la mejora de la calidad de vida orientan también las políticas sociales municipales, que son políticas de vanguardia que van más allá de lo meramente asistencial. Sirvan como ejemplo programas como Salamanca Acompaña, dirigido a las personas mayores de 60 años que se sienten solas y que encuentran en esta iniciativa una puerta abierta para su retorno a la vida social. O el proyecto Edades, un programa único en España, en el que desde el Ayuntamiento, en colaboración con la Universidad de Salamanca, ofrecemos un servicio de detección precoz del Alzheimer y de estimulación cognitiva de las personas enfermas, porque estamos convencidos de que en estas enfermedades es vital actuar a tiempo. Esta labor de apoyo a las personas mayores se complementa con la colaboración que prestamos a todas las asociaciones del ámbito de la salud, más de 50 en nuestra ciudad, que ofrecen a las personas enfermas una amplia cartera de servicios más allá de los que presta nuestro excelente sistema de salud. Salamanca es también una ciudad que apuesta por la ciencia y la investigación. Somos, querido rector, la ciudad del talento, con 2 universidades, una de ellas 8 veces centenaria, y centros de investigación punteros. Los jóvenes investigadores a quienes apoyamos con becas para que se queden a trabajar en Salamanca o a quienes atraemos para que desde el extranjero vuelvan a Salamanca a desarrollar sus proyectos son una pieza esencial para construir la ciudad que soñamos. Y en ese sueño está también la posibilidad de convertirnos en la futura sede del Spain Biotech Institute, un instituto de investigación multidisciplinar que integrará la inteligencia artificial con otras ciencias, con especial foco en las neurotecnologías. La Junta de Castilla y León ha presentado la candidatura de nuestra ciudad ante el Gobierno de España para que a través de los fondos Next Generation este instituto se implante en Salamanca. Nuestro aval es la experiencia de la universidad, la calidad de nuestros investigadores y el apoyo institucional. Desde hoy, también lo será la celebración de esta cumbre, que nos sitúa en el foco mundial de los avances en la prevención, el diagnóstico y el tratamiento de las enfermedades neurodegenerativas. Finally, I just want to wish you all that, as well as you may get a great benefit from these working days, you also may find the chance to enjoy the city of Salamanca, the charm of its heritage buildings, the taste of its gastronomy, and the warmth of our citizens. Thank you very much. Muchísimas gracias.
Tiene la palabra don Ricardo Rivero, rector magnífico de la Universidad de Salamanca.
Su Majestad, la Reina de Suecia, Su Majestad, la Reina Doña Sofía, excelentísimo señor embajador de Suecia, señora delegada del Gobierno en la Comunidad Autónoma de Castilla y León, ilustrísimo señor alcalde de Salamanca, señora subsecretaria del Ministerio de Sanidad, Consumo y Bienestar Social, autoridades excelentísimas e ilustrísimas, ponentes e investigadores de este encuentro Global Summit Neuro 2020-2022. Señores y señoras, bienvenidas al Paraninfo, lugar de esperanza y de saber y faro del conocimiento europeo e iberoamericano, en este edificio de escuelas mayores donde la dignidad de la persona y los derechos humanos ya se explicaban hace 500 años. El concepto contemporáneo de individuo, agente de su propio destino y respetable en cuanto tal, no puede entenderse sin las aportaciones de la Escuela de Salamanca. Gracias por elegir nuestra universidad para la celebración de este encuentro. Gracias a quienes han contribuido con su esfuerzo a la organización. Gracias a la Fundación Reina Sofía y también a la Fundación 100 y al Ayuntamiento de Salamanca y al Instituto de Salud Carlos III por su patrocinio. La relación histórica del estudio con la corona se demuestra de nuevo hoy. Nos alegra sobremanera que presidáis nuestros actos, Majestad, expresando con vuestra presencia un elocuente compromiso con la investigación, la ciencia y las contribuciones universitarias al progreso social, tarea que tomamos muy en serio en la Universidad de Salamanca. Porque estamos convencidos de que la ampliación de las capacidades humanas es nuestra primera razón de ser. Crear y acrecentar las capacidades para el desarrollo es nuestro cometido, tal y como ha expuesto la filósofa Premio Princesa de Asturias en 2012, Marta Nussbaum. Y este congreso orienta todas las fuerzas de la investigación a proteger las capacidades más necesarias en tal propósito, que son las mentales, las del cerebro. Podemos mantener nuestra identidad personal con pérdidas o deterioros de otros órganos, pero no somos nosotros mismos de idéntica manera si nos falla el cerebro. Comprender el deterioro cerebral y nervioso es presupuesto de la solución de estas patologías, una labor constante que agradecemos a investigadoras e investigadores que trabajan sin descanso en los laboratorios, en los institutos de investigación de la Universidad de Salamanca, en el Instituto de Neurociencias de Castilla y León, en los institutos conjuntos junto al CSIC en la ciudad de Salamanca y en cada uno de los departamentos y grupos de investigación que contribuye en esta línea, sin olvidar al Instituto de Integración en la Comunidad. Todos los grupos de investigación de la Universidad de Salamanca deben ser reconocidos y agruparán sus fuerzas en el proyecto al que ha hecho referencia el ilustrísimo señor alcalde, que tanta energía y tantos esfuerzos está desplegando en nuestras potencias investigadoras para acrecentar y prolongar la calidad de vida. Cada vez nos mostramos más convencidos de vivir en una integración plena entre mente y cuerpo, interactuando en todo un entorno ambiental con un cerebro húmedo, en la expresión de la también princesa de Asturias en el año 2019, Siri Husbeth, recogida en su obra Los espejismos de la certeza. Una enseñanza constante de sentido común recuerda que lo que natura nos da debemos protegerlo, mantenerlo y revivificarlo. El cuidado del bienestar físico contribuye al mental, pues al fin mente y cuerpo no representan un binomio en realidad. Estos días en Salamanca demostraremos nuestra capacidad de cuidar el cuerpo y la mente. Paseen ustedes por las aulas de este edificio y las hermosas calles de la ciudad, donde tantos pensadores aportaron su inteligencia. Disfruten de la excelente oferta cultural, que es muy necesaria para un cerebro sano. Y sepan siempre que les estamos agradecidos y les damos la bienvenida. Aquí, donde se admira la sabiduría, donde leer y escribir es un hábito sempiterno, y aplaudimos a quienes se comprometen con el desarrollo del conocimiento en esta Universidad de Salamanca, que cuenta, como he dicho, con excelentes grupos e institutos de investigación, también hoy aquí representados, los mejores entre nosotros. Gracias otra vez, majestades.
Tiene la palabra don Pascual Sánchez y don Adolfo López en representación del Congreso Global Summit Neuro 2020-22.
Majestad Reina Silvia de Suecia, Majestad Reina Sofía, delegada del Gobierno, subsecretaria de Sanidad, rector magnífico, excelentísimo señor alcalde. La enfermedad de Alzheimer es la única de las 10 principales causas de muerte que no tiene tratamiento, no se puede prevenir y no se puede ralentizar. Se prevé que su prevalencia en el mundo se va a triplicar y en el año 2050 se va a alcanzar la cantidad escalofriante de 150 millones de enfermos. Los trastornos neurodegenerativos en general son probablemente la última frontera del conocimiento biomédico y encontrar un tratamiento para la enfermedad de Alzheimer probablemente sea uno de los principales retos médicos al que nos enfrentamos. Este gran desafío precisa, sin duda, una colaboración estrecha de científicos de diversos ámbitos y disciplinas. El Congreso de Cibernet y la Fundación 100 por naturaleza se ha caracterizado por su mestizaje. En este congreso siempre han confluido básicos, clínicos y las distintas áreas de Cibernet, enfermedades neuromusculares, la enfermedad de Alzheimer, la enfermedad de Parkinson, han realmente enriquecido, dando pues una riqueza de ideas y generando una polinización cruzada del que han surgido proyectos transversales y con carácter translacional. Un ejemplo próximo es el Consorcio Nacional de Genómica de Demencias, de GESCO, que se ha incubado en estas reuniones y que ha convertido a nuestro país en una de las zonas más dinámicas en el campo de la epidemiología genética de las demencias. Iniciativa que se ha trasladado a otras entidades como la ELA y más recientemente la parálisis supranuclear progresiva. Esta es una edición muy especial. Celebramos la Global Summit on Neurodegenerative Diseases. Es una edición que está dentro de una trayectoria de nuestro congreso. Ha sido una referencia con el apoyo de la Fundación Reina Sofía, con Su Majestad la Reina. Se ha convertido en un punto de encuentro regular para los distintos investigadores y que ha permitido que se establecieran exitosas colaboraciones científicas. Este año es especial ya que fue suspendido en el año 2020 y este año tenemos muchísima ilusión. Ahí tenemos la presencia de investigadores tanto básicos como clínicos que nos van a hablar de un amplio espectro de temas, desde los últimos avances en los mecanismos de estas enfermedades hasta la aplicación de la medicina de precisión en la práctica clínica. En estos tiempos la palabra celebración adquiere un significado mucho más pleno y esperamos celebrar un gran congreso en la hermosa ciudad de Salamanca. Muchas gracias.
Majestad Reina de Suecia, Majestad Reina Sofía, excelentísimo señor alcalde de la ciudad de Salamanca, excelentísimo rector magnífico, excelentísimo señor embajador del Reino de Suecia, excelentísima subsecretaria de Sanidad, autoridades, colegas, señoras y señores. Como han dicho anteriormente, este es un Global Summit largamente esperado y que por motivos conocidos ha debido de retrasarse durante unos años, pero esta circunstancia, quizá desgraciada en otro momento, nos ha permitido organizarlo en torno a una fecha muy especial como es el Día Mundial de Conmemoración en torno a la esclerosis lateral amiotrófica, una enfermedad neurodegenerativa que, como ustedes saben, es una enfermedad que afecta a una proporción más pequeña de pacientes que otras enfermedades neurodegenerativas, pero que es realmente devastadora. Todos los años en nuestro país más de 2.000 personas sufren esta enfermedad. El programa 3 de Cibernet, junto con la Fundación 100, ha configurado un espléndido programa científico con un ramillete de investigadores que estoy seguro que es el mejor presagio de esperanza para los enfermos y para sus familias por los logros que seguramente van a acontecer en los próximos días durante la celebración de las sesiones científicas. Este ambiente en el que nos encontramos, un ambiente cargado de tradición académica, con los mimbres humanos que se van a reunir aquí, estoy seguro que van a dar lugar a esta cristalización de esperanzas para los pacientes. No quisiera terminar sin agradecer, como director científico de Cibernet y en nombre de la comunidad científica, a Su Majestad la Reina Sofía su apoyo constante e incansable para apoyar la investigación, no solo en la enfermedad en la ELA, sino también en todas las enfermedades neurodegenerativas. Es un estímulo para todos nosotros. Este día, primer día de verano, un día realmente primaveral con el que Salamanca nos ha acogido, estoy seguro también que es el mejor presagio para poder disfrutar de esta ciudad cargada de tradición y también de modernidad, como nos acaban de decir. Gracias.
Se levanta la sesión. Declaro inaugurado el Congreso Global Summit Neuro 2020-22. Muchas gracias. In short. Yes.
Majestad, Reina de Suecia, Majestad, Reina Sofía, Señora Delegada del Gobierno, Subdelegada de Sanidad, Rector magnífico de la Universidad de Salamanca y excelentísimo Alcalde de Salamanca. Muchas gracias por acogernos en Salamanca para celebrar este congreso. En el marco del acontecimiento Excepcional Interés Neuro 2022, se va a proyectar el vídeo Inteligencia Natural, cuya finalidad es la visibilización de las enfermedades neurodegenerativas y el apoyo a la investigación en estas enfermedades. Este vídeo ha sido realizado por el encargo de la Fundación Reina Sofía.
Vídeos hechos con deepfake, escenas para películas, conciertos con hologramas, grandes avances en inteligencia artificial. Es curioso lo que es capaz de hacer la inteligencia artificial. Asombroso. Por ejemplo, que yo esté aquí hablando con vosotros. Aunque fallecí hace más de 30 años padeciendo una enfermedad neurodegenerativa. Y si además de avanzar tanto en inteligencia artificial, investigamos más nuestra inteligencia natural, quizás así podamos vencer enfermedades que afectan a nuestro cerebro, como el Alzheimer, ELA, Huntington, o como la que yo sufrí, el Parkinson.
A continuación, es un placer para mí presentar a un magnífico investigador y amigo, que es el doctor Fernández Chacón, director del Instituto de Biomedicina de Sevilla y miembro de Cibernet.
Hello. Her Majesty Queen Silvia of Sweden, Her Majesty Queen Sofia, Spanish government representative in Castilla León, Spanish Minister of Health representative, President of the University of Salamanca, Mayor of the City of Salamanca, authorities, dear colleagues and friends, ladies and gentlemen. It is a great honor and a great pleasure for me to introduce our keynote speaker, Professor Thomas Südhof, who is the Avram Goldstein Professor and Professor of Molecular and Cellular Physiology, Neurosurgery, Neurology, Psychiatry at the School of Medicine of Stanford University and investigator of the Howard Hughes Medical Institute. Professor Südhof was born in Göttingen, Germany. He studied medicine and obtained his doctoral degree at the University of Göttingen and performed his doctoral thesis work at the Max Planck Institute for Biophysical Chemistry with Professor Victor Whitaker on the biophysical structure of secretory granules. After that, and with the idea of acquiring a more clinically oriented scientific training, he moved to the University of Texas Southwestern Medical Center in Dallas, to the laboratory of Michael Brown and Joe Goldstein. There, Thomas Südhof elucidated the structure, expression, and cholesterol-dependent regulation of the LDL receptor gene. Soon afterwards, Brown and Goldstein were awarded the Nobel Prize in Physiology or Medicine. In 1986, Tom Südhof started his laboratory as a Howard Hughes medical investigator at the University of Texas Southwestern Medical Center in Dallas, where he worked until he moved to Stanford in 2008. In 1986, he was fascinated with the idea of studying neurotransmitter release. At that point, though, not a single synaptic component had been characterized at the molecular level. Now, more than 3 decades later, a molecular framework that plausibly explains neurotransmitter release in molecular terms has emerged, in great part thanks to the major contributions of Thomas Südhof. Among those key contributions are the discovery of the molecular mechanisms underlying membrane fusion during synaptic vesicle exocytosis and the elucidation of how calcium sensing is mediated and controlled neurotransmitter release. His contributions has been published in a very impressive collection of more than 630 highly meaningful and data-rich papers in the best scientific journals. He certainly is one of the most renowned players of contemporary neuroscience. More than 25 years ago, his laboratory discovered neurexins and neuroligands, proteins that later on turn out to be synaptic adhesion molecules altered in autistic patients. Thomas Südhof studies how synapses are formed to enable the establishment of the specific connections that build up a neural circuit. His research aims to contribute to our understanding of how synapses become dysfunctional and degenerate in neurological and neuropsychiatric disorders. Thomas Südhof is a member of the National Academy of Science, the Institute of Medicine, the Royal Society of the UK, the German Leopoldina Academy of Natural Science, and the American Academy of Art and Sciences. He's the recipient of multiple awards, including the National Academy of Science Award in Molecular Biology, the Bristol-Myers Award in Neuroscience, the Kavli Award in Neuroscience, the Lasker-Debacki Medical Basic Research Award, and the Nobel Prize in Physiology or Medicine. Thomas Südhof has impacted the career of many young investigators and trainees that, as indeed was my case, had the privilege of training in his lab, benefiting enormously not only from his mentorship, creativity, and scientific talent, but also from his daily example of discipline, hard work, independence, and commitment to excellence in fundamental science. First time Thomas Südhof came to Spain, I think for scientific reasons, was in 1997 when he visited the Department of Medical Physiology and Biophysics of the School of Medicine in Seville. Since then, he has maintained a lively scientific relationship with Spain. Every year, in our Institute of Biomedicine of Seville, we name a scientist as an IBIS Distinguished Investigator to recognize her exceptional contributions to the advancement of biomedicine. Thomas Südhof, in 2015, was the first IBIS Distinguished Investigator. At the city of Baeza, the UNESCO World Heritage Site in Jaén, the International University of Andalusia organizes every year the workshops Currents, Trends in Biomedicine. Thomas Südhof has served as organizer and speaker in several workshops. Destiny decided that in 2013, he was indeed driving by car into Baeza when he got a surprising call communicating him the awarding of the Nobel Prize. That was a great day. That was a lot of fun. Professor Südhof, dear Tom, we're really happy and honored of having you here with us today in Salamanca. Thank you very much for coming. Please join me to welcome Professor Thomas Südhof.
I feel kind of embarrassed to be standing here as though I'm going to preach. I'm not. Okay, so Her Majesty, Queen Sylvia of Sweden, Her Majesty Queen Sophia, dear representatives of the local university, of the government of Spain, and especially dear colleagues, dear friends who came to this event. I would like to thank the organizers for giving me the opportunity to talk to you here today in Salamanca, which is one of the oldest universities of Europe, maybe of the world. The city of Unamuno, whose spirit, I suppose, is still with us in many ways, and who is still, I think, a guiding inspiration for many here. It is truly an honor for me to be here and to be the first speaker in this wonderful symposium. So thank you very much for having me. What I would like to do today is tell you a bit about our personal research on Alzheimer's disease, or rather on a basic biological question that relates to Alzheimer's disease. But before I go into this, let me just, as I have to, disclose my potential conflicts of interest. I am an advisor or board member of a number of companies that are listed here. Of these, most important is probably my work as a member of the board of directors of Sanofi, which is one of the biggest pharmaceutical companies, where I am a general scientific member of the board and enjoy working with many European institutions on trying to advance drug discovery for a wide variety of diseases. Nothing I will talk about today relates to any of the work I do with companies. I enjoy working with companies. Some of the companies I work with are focused on neurodegenerative diseases such as Electra. But the work I'm going to discuss today is independent or different from my consulting and biotechnological work as an advisor and founder of companies. What I want to do today is first talk to you by way of a broad introduction to what I conceive from my personal perspective to be the problem of Alzheimer's disease. This is going to be very general and abbreviated to give you some taste for how I personally think about the challenges we face. And then I'm going to tell you about a specific project, just one project, for the rest of my presentation. So let me start at the beginning, always good. This is truly an abbreviated discussion of Alzheimer's disease, and I apologize for those of you who are more knowledgeable about the disease if it's too simple. But I wanted to give a background for why we are doing the experiments that I'm going to present to you in the second part of my discussion. Alzheimer's disease, like all neurodegenerative diseases, is a very tragic disease. It was named after Alois Alzheimer, who discovered the disease and specifically characterized this first patient, Auguste Deter, who actually had a mutation in one of the key genes that I'm going to discuss called presenilin-1. Alzheimer's disease is tragic because it is progressive, slowly progressive, and basically robs a person of their mental abilities. It starts as mild cognitive impairment with short-term memory deficits, but then slowly progresses towards a complete loss of all cognitive and eventually all brain functions. It is truly a tragic disease to have, both for the patients and for the people, for the families, for everybody around the patients. And it is a major problem. What you see here is a summary of the incidence in the United States. 11% of all people above 65, which unfortunately includes myself, have Alzheimer's disease. This is enormous. If you go, it's an age-dependent disease. It's a disease of aging. Actually, like many diseases, the probability of having it increases dramatically as we get older. Above the age of 85, more than a third of all people suffer from the disease. This is enormous. This is millions and millions of people. This is a problem that we need to deal with as a society. As another illustration, what this illustrates is the changes in the select causes of death in the last 20 years. And you can see that the only major increase is in Alzheimer's disease. And this is because people are getting older. They are allowed to get older. We have the positive experience in our times that our life expectancy increases, but this comes at a cost. because with increasing life expectancy, there's also increasing risk of disease, not only, but including Alzheimer's disease. This is truly an epidemic that, as a society, we need to deal with. We need to find ways of attacking this problem. When you look at the brain of a patient with Alzheimer's disease, as you can see in this illustration here, there's a tremendous shrinkage because the cells in the brain, in particular the nerve cells, die. This is particularly strongly observed in areas of the brain such as the cortex and hippocampus, but eventually affects the entire brain. As a result, the ventricles become bigger, they expand, and the brain becomes much, much smaller. This is, as you can see, a terrible disease. The brain shrinks because nerve cells die. When you look at these nerve cells at the brain, in a microscope, as already observed by Alois Alzheimer, there are several very typical pathological features. What you see on this slide on the left is a normal brain, very schematically. On the right, an Alzheimer's disease brain. And what you can see is that there's features called amyloid plaques, aggregates of protein that accumulate and are typical of the disease. There are also other types of aggregates called neurofibrillary tangles. The combination of those two features are characteristic of Alzheimer's disease. In addition, there is lipid accumulations in glia cells. There's a massive activation of microglia, a neuroinflammatory response. There's astrocytosis, a proliferation and activation of astrocytes, another type of glia. The brain changes dramatically as it is basically dying. Plaques develop, in fact, decades before people have any symptoms. You can already observe the accumulation of these pathological features in patients who are not yet diseased clinically very early on. And as they appear, slowly the disease prepares itself to become manifest. And these plaques are largely composed of a little peptide, a little protein called A-beta that may be toxic for neurons. But at least 4 pathological processes appear to drive Alzheimer's disease pathogenesis. These 4 pathological processes are illustrated here very schematically. First of all, as I already mentioned, is the A-beta amyloid accumulation, the formation of plaques that happens long before the disease becomes manifest. Second are the neurofibrillary tangles that occur later. Third is the neuroinflammatory response, the microglial activation. And fourth, there's an endolysosomal dysfunction that leads to a change in lipid metabolism and cholesterol homeostasis. And those 4 processes together somehow lead to synapse dysfunction, to microglial and astrocytic activation that together cause the neurons to die, resulting in cognitive impairments. This is a very abbreviated, schematic view of what happens in Alzheimer's disease. What is the cause of these pathological processes? We don't know. And one of the things I really want to communicate here today is that we do not understand what actually happens in the disease. We have no idea, really, why people get sick. The biggest progress, arguably, at least in my opinion, has been in human genetics, in an understanding of the genes that predispose to Alzheimer's disease. And these genes suggest, indeed, that there are multiple parallel pathogenetic mechanisms in the disease. Let me present to you a very abbreviated genetic landscape of the Alzheimer's disease process. First of all, very rare mutations in 2 types of genes called APP and presenilin invariably cause Alzheimer's disease. This is what was observed in Alois Alzheimer's first patient, It is very rare, but people who have this disorder, these changes, these genetic mutations, always get Alzheimer's disease. It is very, very tragic. Second, there's a very common polymorphism, genetic variation, in an apolipoprotein called ApoE that strongly predisposes to Alzheimer's disease. Specifically, the ApoE4 variant predisposes. And anybody with an ApoE4 allele above 90 almost guaranteed to have Alzheimer's disease. This is not a minor effect. This is a huge effect. And the frequency of these alleles in the populations are not low. They're above 10%. This is indeed the most common genetic predisposition to Alzheimer's disease. And finally, there are less common genetic variations that predispose to Alzheimer's disease, in particular in a gene called TREM2, genetic variations that are thought to primarily be active in innate immunity, such as microglia, in cholesterol metabolism, and in vesicular traffic. This is enormous progress, but here are the problems. Knowing the genes that are involved in Alzheimer's disease in itself doesn't tell us much. It doesn't tell us how the disease actually develops. In today's science, there's a lot of genetic information. There's a lot of presentations that are called Go analyses. There's a lot of apparently definitive studies. But in the end, the fact is that we don't know what most genes actually do. We have assignments, function assignments, that are only very, very limited. We don't even know where these genes act and where their dysfunction is important for diseases. And in fact, in my scientific presentation a little bit later on, I will tell you about how we address this problem for one particular set of genes, presenilins, that, as I mentioned just now, are among the most important genes predisposing to Alzheimer's disease. Let me explain this a little bit more. So presenilins are in some ways functionally well characterized because they're known to be a subunit of an enzyme that is called gamma secretase, a protease. So in some ways we know the function. And in fact, one of their substrates is APP, the other gene that I mentioned in which mutations predispose or cause Alzheimer's disease. And the way this works is illustrated here in this very schematic slide where you see an APP protein in the membrane, and it illustrates how two different enzymes cut this protein into pieces, liberating a middle yellow fragment called A-beta, the same A-beta that forms the plaques. And what you can see on the right side of this illustration is how it forms these plaques. So this is central to Alzheimer's disease pathogenesis because we know that the plaques are characteristic of the disease. The nature of the presenilin and APP mutations suggests that these mutations shift APP cleavage by gamma secretase and thereby alter production of A-beta. And that this creates A-beta forms that aggregate. And let me explain this a little more detail because this is important in understanding the thinking about Alzheimer's disease. APP is a large protein, and the A-beta fragment is just a tiny piece of it. But all mutations in people that cause Alzheimer's disease 100% of the time, as illustrated here, are within that little piece that encodes A-beta. It's suggesting that this is really where the action is in Alzheimer's disease. Very differently, in presenilins, which are membrane proteins that I mentioned already, are the subunits of the gamma secretase protease. The mutations are all over the molecule. However, those mutations too cause a change in the production of A-beta from APP. And together, these findings that are dating back about a decade or so led to what's called the amyloid cascade hypothesis to try to find a conceptual framework that might explain the disease. And this is illustrated here. You don't need to read through this in detail because it's too much detail. We don't need to go through this. The point here is, the idea is that liberation of A-beta causes aggregation of A-beta, and this aggregates, kill nerve cells, and that's why we have Alzheimer's disease. That's the idea. Okay. Now, with this idea in mind, many drug companies started to develop drugs against A-beta, to treat Alzheimer's disease, to develop drugs that would actually prevent the formation of A-beta or would clear the brain of A-beta. These drugs were developed with investments of many, many billions of dollars throughout the world, in Europe, in the United States, in other countries as well. They were investigated in multiple clinical trials. And they come in two different flavors: small molecules that inhibit the formation of A-beta by blocking the enzymes that create A-beta, or antibodies against A-beta that clear the brain of A-beta. After billions of dollars, I think it's safe to conclude that all these attempts failed. There has been nothing at this point. It turns out that if you block A-beta production, you actually hurt patients more than you help them, for unknown reasons. If you remove A-beta with antibodies, it works extremely well. You can show that the patient's brains no longer have plaques, but the patients aren't really doing better. In only one trial out of many by the company Biogen in Boston, there was a tiny effect, positive effect. It was so tiny that everybody in the field doubted its utility, but the FDA approved the drug nevertheless, called Aduhelm. However, there's been no uptake by the community because the effect was so small that few people actually want to take it. So, why didn't this work? Why is it that after all this effort, after we had such a clear perspective, we have made so little progress? And here is what I would like to convince you of. My personal opinion, because we don't really understand what's going on. There is no real evidence that A-beta itself is causing AD. I think personally it's likely a cofactor, a component. But in itself, you can't blame everything on it. And we really don't understand the disease itself. We don't actually understand what's going on in the disease. As scientists, we are always admonished to translate our findings, to apply our discoveries. Many people in the public think that as scientists we are a little stupid and we don't know how to basically turn our discoveries into drugs. Let me tell you, that's not true. You just can't translate anything if you don't have any discoveries, basically. It's as simple as that. And premature translation leads to a huge waste of money. Clinical trials, as I can tell you from my experience with working with drug companies, are insanely expensive. And they are also a burden to the patients. We should really be careful how we spend our money in order to make progress in dealing with this devastating disorder. So when you look at this, we just don't know how the various pieces that characterize Alzheimer's disease at this point fit together. Our understanding at this point is descriptive. It's not mechanistic. What we need is a fundamental translational research that enables us to understand the biology of the components, that enables us to understand why certain gene changes, mutations, actually make patients sick. And in order to do that, we need to understand what these genes actually do. I often encounter in Europe the opinion that we understand a lot about the brain in general, and that computer models will allow us soon to understand the brain and its diseases, so that we don't need animal experiments in future, and that at some point we can reduce everything into some kind of theoretical contextual AI sphere. I think this is a total illusion. I don't think we actually understand the brain at this point. I don't think we will understand the brain in my lifetime. I don't think we will understand it even in the lifetime of my children, who are young. So The brain is very complex. No computer, even at this time, understands a simple cell, just one cell. How is it possible to understand something as complex as the brain, that is more complex than a computer? Okay, it's just, there needs to be a lot more time. And this is a personal opinion. As an aside, I think animal experiments are the only way we will ever be able to actually model brains in the near future, in the next 50 years. And I fear a bit, as a European, although I've lived most of my life in the United States, that Europe will fall behind because we are abandoning approaches that are essential for studying the biology of the brain and for studying its diseases, for studying mechanisms, for identifying treatments, for actually helping people. Let me now turn to the scientific part of my presentation. Maybe that was a bit of preaching after all. I'm sorry. And tell you about one particular project, one particular experiment that we've done based on our view that we need to understand the biology of the key genes that drive Alzheimer's disease. And in order to tell you about this, I first need to introduce you to our approach, how we study this. What we do in these experiments is we study human neurons. And in order to make human neurons, we use embryonic stem cells or iPS cells, which are stem cells, and we transdifferentiate them into neurons in culture using expression of a gene called NGN2. With this approach, we can make neurons within a few weeks. We co-culture the neurons with mouse glia after a few days. So we need animal experiments, of course, because without glia, neurons can't survive. They don't do well. They don't form synapses. They just degenerate. We then add gamma secretase inhibitors to study specifically the role of gamma secretase in neurons, or we make mutations in presenilin, the Alzheimer's disease gene, in these neurons. And we finally analyze them in depth after about 6 to 7 weeks. Using this approach, we can suppress gamma secretase activity and test what is the specific function of gamma secretase in a neuron after it's been born, after it's been made, not during development, but later on, as a way of approaching the question of what might its dysfunction mean for Alzheimer's disease. This works because you can see here, we suppress the total A-beta concentration, although we changed the A-beta 42 to 40 ratio for the aficionados. We increase the uncleaved APP, because APP is a gamma secretase substrate, and we also increase the uncleaved variants of other proteins that are in neurons or synapses, such as neurexins, as shown here. When we mutate presenilins, as shown here, and specifically presenilin 1, we have the same phenomenon. We abolish presenilin 1, obviously, and we increase the levels of the uncleaved APP fragment. So this gives us a toolbox, two independent methods to chronically suppress gamma secretase activity in human neurons, which happens in patients who have, unfortunately, mutations in presenilin 1 and therefore will develop Alzheimer's disease later on in their lives. This is the approach. So what is the effect of this change on the actual neurons? Surprisingly, the effect is extremely selective. There is no effect on the actual shape of these neurons, on their ability to develop dendrites or axons, as shown here. The axon length is normal, the dendrite length is normal, their branching is normal, their arborization is normal. There is, however, a small change, a small decrease in the size of the soma, of the cell body of the nerve cells. So these nerve cells are doing overall very well. It's not essential. They don't need this, in fact. What happens to their synaptic connections? The connections between nerve cells that enable them to talk to each other, which I have been studying for most of my scientific career. Well, it turns out that surprisingly, as seen on this slide here, there's an increase in synaptic connections morphologically. What you see here is that when you measure the density of synaptic connections by immunocytochemistry using a marker that's specific for the presynaptic site, there's an increase. If you measure it using a marker that's specific for the postsynaptic site, there's an increase. And if you only measure the puncta that have both markers as the most rigorous approach, you can see that that increase is even more robust, demonstrating that the gamma secretase inhibition, chronic gamma secretase inhibition, causes an increase in the density of synaptic connections. This is mirrored by an increase in the amount of synaptic protein PSD95, as shown here. So, somewhat surprisingly, you don't actually see a loss of synapses. Here you see an increase of synapses. Now, mind you, these are in culture. These are only a few weeks, 6-7 weeks, as a model for a human disease that doesn't happen until the 5th or 6th decade. There's huge limitations here. So the fact that there's no loss of synapses at this point doesn't necessarily mean that there wouldn't be any later on. The question here really is, what is happening to synaptic function, to the actual process by which the neurons communicate with each other? Is that also increased, or is there something else going on? And what is happening is illustrated here. The actual synaptic transmission is decreased. As you can see here, this is a robust change. It is measured by electrophysiology, where you measure the postsynaptic strength, the postsynaptic signal as a function of an action potential. And you can see here that there's about a 40% suppression of the strength of the synapses with this manipulation. So we have more synapses, but the synapses are just not as good. They're impaired, they're dysfunctional, which fits with Alzheimer's disease in the sense that Alzheimer's disease may be a synaptic disorder, a synaptic impairment. Is this an acute effect? Does this happen as soon as you block gamma secretase, or does this sort of chronically develop secondarily? To measure this, we examined treatments of only 24 hours or 48 hours, and as you can see at 24 hours, there's a trend at 48 hours. It becomes significant, but slowly so. So this is not acute effect. This is something that happens after gamma secretase inhibition, that something happens that then causes synapses to become more numerous, but impaired, dysfunctional. Why is synaptic transmission impaired? Now this goes a little bit into the nitty-gritty of synaptic transmission. For those of you who don't love synaptic transmission, just tune out. I love synaptic transmission. I always find it amazing how neurons talk to each other at synapses, how fast and precise the process is. And what you see here is that when you just analyze the spontaneous responses in synapses, there's not much of a change. So the actual spontaneous activity is unchanged. But if you look at evoked synaptic transmission, at the response to an action potential, and you measure the probability that an action potential will cause neurotransmitter release, as shown here, by measuring either the paired pulse ratio or the coefficient of variation, shown here on the right, you will find that what happens upon chronic gamma secretase inhibition is that there's a major impairment in the release probability. And that is illustrated here in this slide on the left with the paired pulse ratio, and on the right with the coefficient of variation. I'm not going to go into the details. This goes into the arcana of synaptic transmission that are not relevant for today's discussion. Suffice it to say that what happens then is that chronic suppression of gamma secretase, not total inhibition, just suppression to some extent, leads to a change in the synaptic function that causes a proliferation of synapses which are impaired because they can't release neurotransmitter as well as they should normally do. And this summarizes what I just said. So I don't need to say it again. Okay. All this was done with pharmacological inhibition. What about if we do the same thing using a genetic inhibition, as you see in Alzheimer's patient? And basically you get the same result. If you mutate presenilin-1 that inhibits gamma secretase, you get the same impairment of synaptic strength, and you get the same change in release probability as evidenced by the change in paired pulse ratio and the coefficient of variation. And you see multiple clones of presenilin-1 mutants here as a control, because you want to make sure that this is independently reproducible between multiple clones. They all have the same phenotype. What these data thus demonstrate is that chronic inhibition of gamma secretase in human neurons, not in the glia, it's in the neurons, because that's where the presenilin mutation is, increases synapse numbers but decreases neurotransmitter release probability. It makes overall synaptic communication weaker. It impairs it. It makes synapses dysfunctional. How does this work? To address this question, we used the standard approach nowadays, which is to look at gene expression. The reason for that is particularly pertinent because I haven't discussed this with you, but it's well known that gamma secretase is critical during development in regulating gene expression via the Notch pathway. And because of that, our hypothesis was that gamma secretase inhibition might be acting indirectly via gene expression changes. And so we studied gene expression using RNA-seq. This is bulk RNA-seq for the experts in you. And what you see here is a so-called volcano plot. I'm not going to go through this because you can't even see them. It's impossible with these monitors. Instead, let me just summarize to you. What you see here on the left is the gene expression in the neurons. What you see on the right is the gene expression in the glia, mouse glia. What you see in the neurons here with chronic pharmacologic inhibition of gamma secretase is that virtually all upregulated genes are involved in cholesterol synthesis or cholesterol uptake. This was a huge surprise. Downregulated genes are primarily immediate early genes, which is not surprising because there's a decrease in synaptic function, so there's a decrease in network activity, so there's a decrease in activity. None of this is observed in mouse glia, so this is specific. The same phenotype was detected with a second inhibitor, completely different chemical, demonstrating that this is specific, this is not something that randomly occurs. To show this in a different way, what you see here is the fold change in the neurons, either upregulation or downregulation of genes. The top 20 genes are all cholesterol-related genes. Amazing. Downregulation of genes are more diverse, very different types. This means that clearly what happens with the chronic gamma secretase inhibition is that we are influencing the cholesterol pathway, which is in itself interesting because cholesterol has been implicated in Alzheimer's disease. Just as a demonstration, the mouse glia that also have the pharmacological, although not the genetic inhibition of gamma secretase, have none of this. So gamma secretase only acts in the neurons on the cholesterol metabolism, not in the mouse glia. There are many changes in microglial and astrocytic genes, as you would expect, because that's what the mouse glia is composed of. But there's no functional, coherent pattern as opposed to the neurons. And all of this was validated by quantitative RT-PCR. So when you look at cellular cholesterol metabolism, as shown here, very schematically, what we find is that de novo synthesis of cholesterol in neurons and cholesterol uptake are highly upregulated by this chronic activity. How is this? What is happening? Why is this? Now, there's two potential explanations, at least two potential explanations for this phenomenon. One is that the gamma secretase inhibition causes a direct activation of cholesterol synthesis and uptake gene transcription. If that was so, cellular cholesterol levels should be increased. Another explanation is that this enhancement of cholesterol synthesis gene expression is indirect because there's a suppression of cholesterol synthesis. And the neurons react to that, a neuron-specific suppression. And if that was the case, the cellular cholesterol levels should be decreased. We can test these two hypotheses simply by measuring neuronal cholesterol. And we did this using Philippine staining that monitors, measures the surface cholesterol in the plasma membrane. And what you see here is that in this case, using two different gamma secretase inhibitors, chronic treatment, there is a massive decrease in cholesterol on the cell surface of the neurons. The similar change is observed in the presenilin mutants. So clearly there's a decrease in cholesterol. And this decrease, in turn, explains why there's an increase in cholesterol synthesis gene expression. To validate this by a different method, we isolated neurons and glia separately by FACS, as shown here, and we measured their cholesterol directly, the total cholesterol, not just the plasma membrane cholesterol. And what you can see here is that in the neurons, again, the total cholesterol is suppressed by chronic gamma secretase inhibition, whereas in glia, there's no change. Replicating the gene expression studies I showed you. So this is consistent with the transcriptome analysis. So the bottom line here is that chronic gamma secretase inhibition causes a decrease in cholesterol levels in the neurons, but not the glia, resulting in an upregulation of cholesterol synthesis genes. And that, in turn, causes a change in synaptic transmission, as we have shown in additional experiments that I don't have time to discuss today. On a personal side note, as Rafa in his very nice introduction mentioned, this is a blast from the past for me because when I was a little bit younger, as a postdoctoral fellow, I worked on cholesterol in the Brown and Goldstein lab, and I worked exactly on these problems. So I'm coming back to my roots here. So this illustrates to you, I hope, what I think is just one tiny part of a large number of experiments we need to understand better the biological basis of what happens in these neurodegenerative diseases. In my last few slides, I would like to end with some remarks on the relation to Alzheimer's disease that might be present here, but also I would like to end with a note of caution. I can't offer a facile and definitive conclusion about how the disease develops. I don't know. I feel completely open to any hypotheses that are out there. I think that A-beta is likely a big factor of the disease, but I don't think in itself it's the solution. But I do want to note that many of the genes that are implicated in the development of Alzheimer's disease are actually related to cholesterol metabolism. That is a pathway that represents a major part of the genetic risk for AD. And I just want to remind you that ApoE is the most important risk gene for Alzheimer's disease. And after all, as I worked on almost 40 years ago now, ApoE is an apolipoprotein whose major function is to transport cholesterol. both to put cholesterol into neurons and to take it out via these two genes. And as it happens, this uptake exclusion pathway via APCA1 that you see here on the left of this slide also represents an Alzheimer's disease risk gene, APCA1, just like ApoE, although by no means as strong as ApoE. In addition to that, vesicular trafficking genes have been implicated in Alzheimer's disease. So there is a strong genetic reason to think that lipid metabolism, and specifically cholesterol, may play a role in the pathogenesis of Alzheimer's disease. How that operates at the level of the neuron and how that relates to microglial activation remains unclear. Moreover, synaptic dysfunction likely is a major contributor to cognitive decline in Alzheimer's disease. At some point, it was fashionable to talk about Alzheimer's disease as a synaptic disease. I don't know exactly what that means, but it's clear that the initial impairment in Alzheimer's disease happens at the synapse, at synaptic dysfunction, and that causes the initial cognitive symptoms before the neurons die and before you can see any of the morphological changes except for the plaques and tangles. And what I show you here is taken from a paper that was published a decade ago, an MRI imaging paper that shows, for example, that in a patient who hasn't developed any symptoms yet but has a presenilin 1 mutation, so that person tragically will definitively get Alzheimer's disease in a few years. You can already see a major change in how the nerve cells communicate with each other. This is a clear precedent to what actually happens in the disease, suggesting again that a change in synaptic transmission that is related to gamma secretase activity may be an integral component of Alzheimer's disease pathogenesis. Moreover, one of the key features, as I mentioned before, of Alzheimer's disease is the fact that it's a disease of aging. In some way, we can feel grateful that it's not happening early in our lives, but it does happen invariably as we age in a larger and larger part of the population. It's an age-dependent disease, which basically means it must be a disease that occurs cumulatively at some point, that happens gradually until it becomes manifest. And a chronic metabolic imbalance fits this temporal profile, consistent with the notion that a lipid dysregulation could be a component of this neurodegenerative process. However, despite these supportive correlations, there's major issues. One is that APP mutations and ApoE4, the major Alzheimer's disease predisposing allele of ApoE, also affect synapses, but somewhat differently from presenilin mutations. We don't yet know how all this fits together. We have pieces, it's like a puzzle. You have a huge puzzle and you're trying to put it together, trying to figure out where the pieces are. We haven't figured it out. The important role of A-beta, which I think is indisputable, even though some in our field now believe that it should be ignored, and of microglia, which is a major point of study right now in Alzheimer's disease, and their relation to synapse, in my mind, remains unclear. This is another piece of the puzzle that we need to reconcile. And finally, there are drugs that suppress cholesterol synthesis called statins that many of us take. How that fits into Alzheimer's disease and why people who have the suppression of cholesterol synthesis apparently do not get a major change in Alzheimer's disease at this point is unclear. So frankly, as I emphasized before, we don't understand how the pieces fit together. There needs to be a much better understanding of the basic biology of the disease process, of the genes, of their functions in specific types of cells in the brain, before I believe we can come to a coherent hypothesis that makes sense. Now, let me end by emphasizing that doesn't mean that clinical trials are hopeless. I think we do need clinical trials even if we don't have a complete understanding. I just think that we need clinical trials that are more exploratory, and we need to know when to stop and move on and test other hypotheses. I am personally involved in several clinical trials via my consulting activity that are targeting microglia. I think this is a good approach, but I also believe, as I just mentioned, that we need to make sure that if those clinical trials don't show a major effect, we should stop them and not just spend more and more money for a small integrative change, but then focus more on getting further ideas, further insights of potential targets. Interesting music. So with this, I would like to end. What I've tried to tell you about today basically is why I believe that as a community, we need to deepen our understanding of what I would call basic translational processes in neurodegeneration, not only Alzheimer's disease. We face a similar problem in Parkinson's disease. We face a similar problem in ALS, Lewy body disease, all this whole gamut of neurodegenerative diseases. I think that clinical trials via drug companies is a useful approach, but I also think that we need to not lose track of the actual biology, which often happens. And finally, most importantly, let me acknowledge the people. The work I discussed today was almost entirely the actual experiments done by Sofia Essayan-Perez, a brilliant MD PhD student who's graduating, with help of Yingsha Zhang, my long-term collaborator and lab manager. And much of the work that we do on human neurons is carried out in collaboration with Marius Wernick and his team at Stanford. And the work I discussed was funded by the National Institutes of Aging in the United States and by the Howard Hughes Medical Institute. I'd be happy to answer any question if there's an opportunity to discuss my presentation. Thank you very much for your attention.
Okay. So, can I have, Tom. So, Tom, thank you very much. Really, a really beautiful and inspiring talk. So the session is open for questions. So whoever is in the room that would like to ask a question, please raise your hand. There's a question there. Maybe you can speak loud. If not, you can maybe come here. Manuel Malmierca. Manuel, why you didn't come here? Okay, come here, probably. Oh, do you have? Okay.
Thank you very much for the beautiful talk and touching our heart and our brain. I think we need people like you that tell our politicians and our scientists how we are and how we are. But I'm not going to go into political issues here. I would like to ask a scientific question. I'm working in hearing, and you may know that a couple of years ago there was a beautiful paper published in Lancet where they found a very direct relationship between hearing loss and the development of Alzheimer's. So the question is, if you know how this is related, what is the causal reason between, that. In hearing loss, we know that there is a deficit in GABAergic inhibition, and I was really struck by your talk, and I wonder if you might have any comment about that. Thank you very much.
It's a great question. I don't know that particular paper. I'm sorry, so I missed it. I can only speculate that as Alzheimer's disease captures the brain of a patient, it differentially affects different types of neurons in different parts of the brain at different times as the disease progresses. And I could easily imagine that parts of the cortex in particular, but maybe also parts of the actual sensory system, may be more susceptible to the process that destroys synaptic function and nerve cells. It is well known that also the sense of smell is impaired early on in Alzheimer's disease, and so that would make perfect sense.
Thank you very much.
Thank you. So we have time for one more question, just in case we have it. Okay.
Thank you very much. My name is Orla Hardiman. I'm an ALS researcher from Dublin in Ireland. And I was intrigued by your data suggesting that there's an early increase in synaptogenesis, but inefficient synapses. And in the ALS field, there's a debate running at the moment about whether some forms of ALS are neurodevelopmental in origin. And I'd like you to comment on that, particularly in those genotypes where we know there's an early genetic variant. And do you think that there could be a developmental role for these types of Alzheimer's disease? Thank you.
It's a great question. If I may rephrase it, the issue here is the fact that genetic predispositions to various neurodegenerative disorders, such as ALS, Parkinson's disease, Alzheimer's disease, happen throughout life, already early in development. So is it possible that people are sort of marked early in development by their genetic changes so that they develop the disease as they age? I see no reason why that wouldn't be possible. It would have to be a change during development that then causes a cumulative damage which then manifests decades later. I think that's entirely possible. I just don't know if there's any evidence for or against. It's a difficult question to test. Yeah. Thank you very much for your attention.
All right, thank you.
Viva